In this video, we will differentiate between infection and disease and examine the manifestations of disease, virulence factors utilized by pathogens, and the stages of infectious disease. As we have previously discussed, infection refers to the entrance and multiplication of pathogens in the host. When pathogens enter and attempt to multiply, the body utilizes a variety of mechanisms to defend itself. We will explore the body's defenses against pathogens in Unit 4. Some pathogens are destroyed by the body. Others are able to survive the body's defenses and continue to multiply. As they continue to multiply, they may adversely affect the body, resulting in disease. Disease occurs when damage or disruption of the tissues or organs caused by the pathogen interferes with normal body function. Disease is also known as morbidity, which is a change from a state of health. When pathogens cause damage to the tissues and organs of the body, which result in disease, it is usually accompanied by a variety of signs and symptoms. Symptoms are characteristics of the disease that are subjective and are only felt by the patient, such as nausea, dizziness, headaches, and pain. Signs are objective evidence. Signs are typically more precise than symptoms. Signs include changes that can be seen or measured in some manner by the healthcare worker and include items such as rashes, fever, vomiting, change in heart rate or respiratory rate, changes in blood counts, among many other items. Some diseases can be identified by a certain set of signs and symptoms. In such cases, the group of signs and symptoms are referred to as a syndrome. Infections may go undetected if they do not present symptoms. The patient may feel healthy, while proper tests would reveal changes, such as changes in white blood cell concentrations. Such infections are referred to as asymptomatic or subclinical. Pathogens are parasitic microorganisms that can cause infection and disease in their host. The term pathogenicity refers to the ability of a microorganism to cause disease. The terms pathogenicity and virulence are sometimes used interchangeably. However, the term virulence actually refers to the degree or level of pathogenicity of the pathogen. These two terms do not provide any reference to the severity of the disease that the microorganism causes, but rather only suggest that the organism is capable of causing disease and at what level the organism is capable of causing the disease. Virulence is determined by the ability of the microorganism to establish infection and disease. Virulence differs between species and among members of the same species. The virulence of pathogens can be considered along a continuum from low to high. Those microorganisms that are less virulent include organisms such as the opportunistic pathogens that are capable of causing diseases in weakened hosts or when they are present in very large numbers. On the opposite end of the spectrum, those that are highly virulent almost always cause disease, even in healthy hosts. The virulence of the microorganism can usually be reflected by the infectious dose. The infectious dose is the minimum number of the microorganism required for infection. The infectious dose is determined experimentally. Generally, the more virulent a microorganism is, the smaller the infectious dose required for infection and disease. For example, the infectious dose of Vibrio cholerae, the causative agent of cholera, is 100 million cells. The infectious dose of Mycobacterium tuberculosis is 10 cells, and the infectious dose of Coxiella burnetii, the causative agent of Q fever, is 1 cell. Thus, the Coxiella are the most virulent of our examples. The same concept applies to viruses. The infectious dose of smallpox is 10 to 100 viruses. The measles virus is more virulent because the infectious dose is 1 virus. The virulence of a microorganism is determined by its ability to cause infection and disease. In order for a microorganism to cause infection, it must first attach to the host and evade host defenses. In order to cause disease, they must damage host tissues or organs. Microorganisms may cause damage by secreting enzymes or toxins, or by inducing a response within the host that results in injury to the host. Characteristics and structures that permit a microorganism to cause infection and disease are known as virulence factors. Virulence factors include adhesion factors, biofilms, antiphagocytic factors, extracellular enzymes, and toxins. A microorganism may employ one or more virulence factors to cause infection and disease. We have already discussed adhesion factors and biofilms in a previous lecture video. We will begin our discussion of virulence factors with antiphagocytic factors. Several structures and tactics are employed by pathogens to evade the phagocytic cells of the body's defense. Such factors and tactics are known as antiphagocytic factors and act to prevent phagocytosis. Some bacterial cells utilize a capsule composed of polysaccharides similar to the host cells to avoid phagocytosis. In this case, the phagocytes fail to recognize the bacterial cell as foreign. The capsules basically act as camouflage, allowing the bacterial cell to go undetected. Cellular structures such as slime layers may be slippery, making it difficult for the white blood cell to engulf the cell. Organisms that utilize capsules or slime layers related to virulence are Streptococcus pneumoniae, Klebsiella pneumoniae, Bacillus anthracis, and Haemophilus influenza. Another mechanism used by microorganisms such as Staphylococcus and Streptococci is to release chemicals known as leukocidins that act to kill the phagocyte by causing its own lysosomal enzymes to be released into its cytoplasm, a process known as autolysis. Some microorganisms are adapted to survive inside the phagocyte following ingestion. There are a number of ways in which the pathogen survives within the phagocyte. Some microorganisms permit phagocytes to engulf them, but once inside, they release chemicals that break down the phagolysosome, thereby releasing them into the cytoplasm, where they may reproduce and at a later point in time rupture the host cell membrane. Other organisms may act to prevent fusion of the phagosome with the lysosome, thereby preventing digestion of the microorganism. This permits the pathogen to reside and reproduce within the phagocyte. When the phagocyte dies, the microorganisms are released to infect other cells. Microorganisms capable of employing this tactic include Mycobacterium tuberculosis, Plasmodium, and Chlamydia. Regardless of the mechanism by which these organisms survive and reside within the phagocyte, this form of antiphagocytosis has special significance because the organisms are provided with a place to hide from the body's defense, multiply, and spread throughout the body. Many pathogenic microorganisms produce and secrete enzymes that break down host tissues. These enzymes are referred to as extracellular enzymes or exoenzymes because they are secreted from the pathogen. Such enzymes may be produced by bacteria, fungi, protozoans, and parasitic elements. Many of the enzymes act to dissolve the organic molecules that act as structural chemicals in the body or by dissolving the host's defensive barriers. Let's examine some of the exoenzymes produced by microorganisms. Mucinase is an enzyme which breaks down mucus that normally acts to protect the mucous membranes. The causative agent of amoebic dysentery utilizes mucinase to digest the protective coat of the digestive tract. Hyaluronidase is an enzyme that acts to break down hyaluronic acid. Hyaluronic acid cements animal cells together. Hyaluronidase is produced by some microorganisms such as Staphylococci and some species of Clostridia. Some of the same species of Clostridia that utilize hyaluronidase also produce an enzyme known as collagenase, which acts to break down the collagen of the connective tissues. Keratinase is an enzyme that breaks down the keratin of skin and hair. This enzyme is produced by those fungi that cause ringworm. Coagulases are enzymes that cause blood to clot and are utilized by some species of Staphylococci, allowing them to hide within the clot from phagocytes. Other enzymes, known as kinases, act to digest blood clots, allowing the organism to escape from the clot and invade the damaged tissues. The many extracellular enzymes, as we can see from the description of their actions, allows them to maintain infections by invading host tissues as they are broken down and by avoiding body defenses. Pathogens may produce and utilize toxins to cause damage to host cells or elicit immune responses that cause damage. Toxinoses are diseases in which the damage has resulted from the action of a toxin. Toxinoses, in which the toxin is spread from the site of the infection by the blood, is known as toxemia. Toxinoses in which the toxin is ingested are referred to as intoxications. Tetanus is an example of a disease in which the toxin is spread in the bloodstream, while botulism is an example of an intoxication. The two types of toxins are endotoxin and exotoxins. We have already discussed the endotoxin found in the outer membranes of gram-negative bacteria. The lipid A component of the lipopolysaccharide of the outer membrane is the only endotoxin. As gram-negative cells die naturally or are lysed by the action of the immune system, the endotoxin is released. Lipid A is of concern because once it's liberated from the outer membrane, it may cause fever, inflammation, diarrhea, hemorrhaging, shock, and blood coagulation. Infections with gram-negative bacteria can be dangerous because of the endotoxin. Gram-negative organisms include E. coli, Salmonella, and Shigella. Exotoxins are toxins that are produced by the microorganism and secreted outside their cells. Exotoxins may destroy host cells or interfere with their metabolism. Several types of toxins may be produced. Cytotoxins are toxins that can kill host cells in general or affect their metabolism. Toxins that specifically target nerve cells are known as neurotoxins, while those that target cells of the gastrointestinal tract are known as enterotoxins. Clostridium tetani and Clostridium botulism are organisms known to produce neurotoxins, while Clostridium difficile and Vibrio cholerae produce enterotoxins. As the microorganism establishes itself and causes disease, the host responds and progresses through distinct stages. In some cases, 5 stages will be observed. The first stage of infectious disease is the incubation period. The incubation period is the time between the initial contact with the pathogen to the time when signs and symptoms first appear. The organism is multiplying and has not yet caused enough damage to permit signs or symptoms. The incubation period will vary greatly between different pathogens. The incubation period may be as short as a few hours or may last for years. The length of the incubation period is dependent on the virulence of the pathogen, the infectious dose, site of the infection, and state of health of the individual. The prodromal period is the time when the patient is experiencing generalized symptoms such as muscle aches, headaches, fatigue, and malaise. The prodromal period is not present for all diseases. The illness stage occurs when the patient experiences signs and symptoms more specific to the disease. At this point in the infection, the pathogen has multiplied and is well established in the host tissues, and the host defenses have not yet fully responded. The decline stage is the period during which the immune system is responding to the pathogen. Signs and symptoms begin to dissipate as the pathogen is eliminated by the body's immune system and/or medications. In some diseases, the signs and symptoms decline very slowly, while in others, the decline of the signs and symptoms may occur very quickly. Some diseases are fatal, meaning that the body is unable to overcome the pathogen and the individual succumbs to the pathogen and to death. The final stage is the convalescent stage. This is the stage during which the body is repairing damage that was caused by the pathogen. You should be aware that individuals can serve as reservoirs of disease during a period of illness. Also be aware that the transmission of a pathogen to another host is not restricted to any particular stage of infectious disease. Diseases like HIV can be spread during the incubation period well before any signs or symptoms appear and can continue to spread throughout the other stages.